Hello, hope you're ready for chapter 14. So we're going to talk about principles of disease and epidemiology. This chapter has a lot of definitions, so most of it you're going to have to remember these concepts, okay? So we are going to talk about some nomenclatures and then related to epidemiology, and then which microbes are part of our microbiota and why, and what keeps microbes away of our body, symbiosis, the Koch's postulate, classifying infectious disease, occurrence of the disease, what is the classification, duration, development of the disease in different reservoirs, contact transmission, vehicle transmission, vectors, and healthcare associated infections. Okay, so we're gonna divide this class in three, where the first part we're gonna talk all the way to Koch's postulate. So let's define pathology, infection, and disease. Pathology is the study of the disease. Etiology is the cause of the disease. So if I say, what is the etiological agent of a specific disease, you know that you're gonna be talking about which microbe is causing the disease, right? Pathogenesis is the development of the disease. Infection is invasion or colonization of the body by the pathogen. And disease is the abnormal state in which the body is not performing normal functions. When we talk about transient microbiota, may be present for days, weeks, or months. That means you may have a microbe living inside your body, not causing disease, that you, for example, contracted in a trip in a different country, in a different place, and that will stay in your body for some time, and then it's just going to be eliminated. The normal microbiota is the permanent microbiota that colonizes the host and does not cause disease under normal condition, and even helps the metabolism of the host. So they're not, most of them are not just not detrimental, but they're also beneficial. Human Microbiome Project analyzes relationship between microbe communities on the body and human health. So there are three different kinds of bacteria that you can find in your body, and that would determine the type of relationship that the host body has with that microbiota. So the name for this, all these relationships is symbiosis, and then they can be of three different types: commensalism, mutualism, and parasitism. So I said three types of bacteria, meaning there are three types of organisms that can be classified as commensal or mutual or parasites. Okay, so when we talk about commensalism, that means one organism benefits, the other one does not suffer. For example, we have Staphylococcus epidermidis and Staphylococcus aureus in our skin as normal microbiota, as we're going to talk in a minute. But we know that they don't cause any harm to our skin unless they become pathogenic. But under normal conditions, they're just there getting some benefit from our skin, but not prejudicing our body. Okay? When we talk about mutualism, then we have a different situation. Both organisms benefit. For example, E. coli in the large intestine. E. coli are super important fermenters. So the fermenters, they ferment sugars that are important in our digestion. So we do benefit from having E. coli in our intestines, and they benefit because they feed from our food and they get protection inside our body. Parasitism is a third situation where one organism benefits and the other one usually gets harmed, right? In this case is where you have diseases happening, right? So in this relationship, you're not talking about normal microbiota bacteria or viruses or any organism. You're talking about organisms that are in the body to cause disease. They are benefiting and we are suffering, okay? So when we talk about normal microbiota, the distribution and the composition of this microbiota is determined by nutrients, physical chemical factors, the host defenses, and mechanical factors. factors, okay? So there is this table in the book that I think is super helpful to understand what point we're trying to make here. So, for example, if we think about the skin, remember that in the skin, these are the principal microbes that we find in the skin. Propionibacterium can cause acne, but it can be just part of your microbiota and not cause any acne. You also have other bacteria like Staphylococcus, Corynebacterium, Micrococcus. Candida is also present in the skin, which is a fungus. So in the comment sections here, we're going to talk about how do we keep microbes away from each one of these regions, okay? And these regions that we're pointing out here are regions where you find microbiota in the human body. So how do we keep microbes out of the skin? First, they don't become residents because they are usually washed away from the sweat and oil glands, because these two components of our skin, these secretions, have antimicrobial properties. Keratin is another physical barrier, and also the low pH of the skin can keep the microbes away. The skin has a relatively low moisture, and that keeps the skin free of microbes. But for example, Staphylococcus, Micrococcus, remember how they're halophiles? The Candida is also halophile. So they don't mind the low moisture of your skin. In this case, they would survive in your skin without any problem. Okay, they thrive in these conditions because they are halophiles. Remember that the sweat that we have on our skin that comes from the sweat glands have lysozyme, and lysozymes kill bacteria. Remember that the oil are usually acidic, and that's why bacteria don't like acidic. They prefer pH a little higher, so a lot of bacteria cannot resist and survive in this condition. The eye microbiota is not that different from the skin microbiota, because the eye is the conjunctive of the eye is a continuation of the skin. Okay, so they have similar microbiota, and what helps also to keep microbes away in the eye is tears that have lysozyme and immunoglobulin, and blinking, which eliminate microbes. Nose and throat, upper respiratory system. So in this part of the respiratory system, you have Staphylococcus aureus, Staphylococcus epidermidis, some other microbes like diphterioids, and Streptococcus pneumoniae, Haemophilus, and Neisseria. These three here, Strep pneumoniae, Haemophilus, and Neisseria, we're going to talk about them later because they can go from the throat if they become pathogenic. If there is some type of disequilibrium in the immune system, they can become pathogenic and enter the blood vessel and cause, among other diseases, meningitis, bacterial meningitis, okay? So in the nose and throat, what helps to keep microbes away and potential pathogens is the fact that the nasal secretions are salty. Again, Staphylococcus always thrives in these conditions because they like high concentration of salt or low moisture. And mucus also keeps microbes away, and the ciliary action of the ciliary escalator of the respiratory system. Remember, the cells have cilia. Those cilia are able to brush mucus with microbes trapped on it out of the way. That keeps them away. In the mouth, remember that in the mouth we have Streptococcus, Lactobacillus, Actinomyces, Bacteroids, Neisseria, Haemophilus, Treponema, Staphylococcus, among others, and Candida. So remember Candida here in the mouth. When we talk about thrush, kids usually have thrush because their immune system is still developing, and this Candida can overgrow and cause that kind of infection in the mouth. What keeps microbes away of the mouth? Obviously, the biting and the chewing and the moving of the tongue, salivary flow, which also has lysozyme and immunoglobulin, are going to dislodge microbes. The saliva contains other also, not just immunoglobulin, but other antimicrobial substances. Remember that you can have a biofilm being formed in the mouth made of these microbes. Large intestine. In the large intestine, remember that you're always going to find the Enterobacteriaceae bacteria. So E. coli, bacteroides, Fusobacterium, those are actually, let's talk about which ones are Enterobacteriaceae. E. coli, Enterococcus, Enterobacter, not Enterococcus, that's gram-positive. Enterobacter, Proteus, Klebsiella, these are Enterobacteriaceae. Enterococcus is gram-positive, but is also present in the large intestine. In the large intestine, you can keep microbes away by the presence of mucus and the periodic shedding of the lining that prevents the microbes from attaching to your intestinal wall. The fact that the stomach is acidic is another way to keep microbes away. And if you have any type of food contamination, then diarrhea is going to flush these microbes out of the large intestine. In the urinary and reproductive system, these are the bacteria that you can find. So you're going to see a lot of bacteria here that are similar to large intestine and similar to skin, because reproductive system is and urinary system are a continuation of the skin. And at the same time, the urinary system and the reproductive system are close to the anus, right? So that is when you're going to see some mixtures there. How do you keep microbes away? In the vagina, there is a high acidity due to the presence of lactobacillus that produce lactic acid, and this is what keeps most of the microbes away. The mucus secreted also in the lining of these reproductive organs will help to keep microbes away, and shedding of the lining, for example, of the uterus is going to help that. The urinary flow is going to help to mechanically remove microbes, and cilia and mucus also helps to expel microbes from the cervix of the uterus into the vagina, and then the acidity of the vagina, like I said, inhibits and kills them. So now let's talk about this. There was a super smart scientist that was called Robert Koch, and he developed a series of postulates that help you identify a pathogen that is causing a disease. So when you talk about these postulates, you need to keep in mind that there are limitations that we're going to talk about when you apply these postulates. So not every case is where you can apply this type of thought, okay? So what is important in the Koch's postulates? The same pathogen must be present in every case of the disease. The pathogen must be isolated from the diseased host and grown in pure culture. The pathogen from the pure culture must cause the disease when it's inoculated into a health-susceptible laboratory animal. And the pathogen must be isolated from the inoculated animal and must be shown to be the original organism. So let's translate this. When we look at this figure, so this is a sick animal, right? This animal got sick, there is some type of infectious disease. So when you collect sample from this animal and plate it, you should be able to grow the microbes that were potentially causing disease in this animal, but you're still not sure which microbes are those. If microbes grow in culture, you can observe them under the microscope. And then you get this pure colony, right, from this microbe, you expand it, and then you inject in a healthy animal. If that animal gets sick, that means those microbes that were in the first animal were the ones causing the disease in the second animal. You should also be able to collect samples from the second diseased animal and grow it in vitro and observe it under the microscope. If microbes here are identical to microbes here, you can say that you know which microbe is causing the disease. This is the cause postulate. There are some exceptions to this postulate, or some situations where you cannot apply the postulates, okay? So we're going to talk about those now. So pathogens that cause many diseases, for example, Mycobacterium tuberculosis, can cause disease in lungs, skin, bones, and other internal organs. So in this case, it would be hard to isolate the pathogen because you don't really know where you would be isolating from, and the diseases are different, right? There are different types of tuberculosis, not just in the lung. So this is the point. Pathogens that cause disease only in humans, for example, Corynebacterium diphtheria and Clostridium tetani, are both microbes that can only cause disease in humans. So you cannot cultivate them and grow them in animals to do the test, right? And you cannot do this test in humans. Microbes that do not grow or do not grow well in artificial media, or what I call in vitro, Treponema, Mycobacterium, Rickettsia, viruses, really hard time growing these organisms in vitro. So that would be hard to use and prove the cause postulate. Okay, so I will talk to you in the second part of the video about chapter 12, or sorry, 14, epidemiology. Bye.